Hello, my name is Megan Wright, and I will be presenting about underdeveloped Cloacal Bursae and age-dependent surfacing postures in diamondback terrapins. Diamondback terrapins occur along the eastern and Gulf coasts of North America and typically live in brackish tidal wetlands. Our study was based on two age groups of juvenile diamondback terrapins at Zoo Atlanta that were being reared as a part of a Head Start conservation program in collaboration with the Georgia Sea Turtle Center on Jekyll Island. Initially, we observed that the adults had a strange surfacing pattern, and we were wondering if this was age-dependent. Luckily, we knew that a new cohort was arriving soon, so we observed the yearlings and then the hatchlings when they arrived to see if there was a difference. We hypothesized that, when yearling turtles are breathing at the surface, they are more likely to spend a larger proportion of time in a vertical position than hatchling turtles. So every week, for 6 weeks, we filmed a section of the tank for about 2 hours. We were only able to take one recording of the older cohort, the yearlings, but they surfaced more frequently than the hatchlings, so we documented plenty of instances. And then the other two co-authors and I rewatched the videos and noted the timestamps for when the terrapins surfaced, when they changed positions, and when they dove back down. So from the time their head was above the water to the time they dove back down was one instance. And then we recorded the proportions of each of the different surfacing postures within that instance. And here you can see our ethogram: vertical, horizontal, ambiguous, and basking. Ambiguous and basking were not counted in our statistical analysis, and they were only there to make sure that we were finding the correct behaviors. To analyze our data, we calculated both the time and percentage of each posture in the whole surfacing instance. Altogether, there were 172 surfacing instances, 100 of which were for the yearlings and 72 for the hatchlings. We totaled these numbers and compared between age groups. We also compared the time and percentages spent in each position within the age groups. We used Mann-Whitney-U tests to analyze the values, and the finding from these will be on the next slide. As you can see, this data is very stark. Specifically the graph on the right, which shows the differences in the percentage of time of each cohort in each position. The yearling data is shown by the dark gray bars, and the hatchling data is shown by the white bars. In the graph, you can see that the hatchling spent a larger proportion of time in the horizontal position, and the yearling spent a larger proportion of time in the vertical position. The findings from our Mann-Whitney-U tests can be seen on the far right, with all of our p-values pointing to the significance of our data. So, we found that the terrapin cohorts were orienting themselves differently at the surface, but then we had to ask ourselves why this phenomenon might be happening. Through a literature review, we found some previous research on Cloacal Bursae, which allow turtles to regulate gas exchange. Essentially, turtles will take water in through the cloaca, which is this whole structure on the left, and then it will go into this small bladder-like structure right here, which is the Cloacal Bursae. And these are the structures that regulate gas exchange. In other amidid turtles, which is the same family as our terrapins, it was determined that Bursae not only assisted with gas exchange, but that they also allow freshwater turtles to take water in through the cloaca as a form of buoyancy control. This means that they take in water to sink and then release it to float. After coming across this information, we were wondering if there might be ontogenetic development in this structure to explain our differences in surfacing posture. So, we requested some museum specimens to loan from both the Museum of Comparative Zoology at Harvard University and the Georgia Sea Turtle Center on Jekyll Island to dissect and see if diamondback terrapins have Cloacal Bursae or not. In the image on the left, you can see the Cloacal Bursae of the red-eared slider, and as you can see, we found wide apertures that led to the thin sac that we saw in the diagram on the last slide. Bursae have been previously described in diamondback terrapins by Pickles in 1899, but when we dissected the adult terrapins, we didn't find them. Instead, we found this remnant structure on the right with divots to bursae, but no cloacal sacs. And we think this may be indicative of an independent evolutionary loss. Diamondback terrapins are one of the only turtle species to live throughout brackish environments, so we hypothesize that the increased salt uptake that would take place during gas exchange renders terrapins unable to use Cloacal Bursae to maintain osmoregulation. But now that we know that diamondback terrapins don't have bursae, this can't explain why we are seeing different posturing behaviors between our two age cohorts. So we do know that adult loggerhead sea turtles can regulate their buoyancy by altering their lung volume, but it appears that hatchlings and juvenile turtles can't actually do this. This study by Mendelson in 1975 reported that turtles younger than 1 month have basically no buoyancy control and that they were unable to dive to the bottom of their tanks. And it's important to note that sea turtles don't have the bursae structure either, and may have similar surface postures as a result. This particular study proposed that buoyancy control was due to underdeveloped anatomical features. We didn't specifically look at buoyancy manipulation in our terrapins, but the loggerhead study explains that juvenile turtles can't physically control a vertical posture for a prolonged period of time. And one of the reasons that hatchlings might have a harder time manipulating their buoyancy is because their shell is not fully ossified yet. It's actually more of a flexible, rubbery structure called fontanelles, and these fuse as the turtle develops. And the rate of ossification and growth has actually been shown to grow more quickly in the anterior shell of diamondback terrapins, which would cause the posterior part of the shell to be less dense, making it difficult to keep the back half of the turtle from floating, which would also help explain our differences in postures. So, hatchling terrapins have been found to spend the first bit of their lives on land, under dense vegetation rather than in the water. And this makes sense from a predation standpoint. Along with a smaller size and a softer shell, hatchling terrapins that are unable to maintain a vertical surface posture also have more exposed surface area to aerial predators from above and aquatic predators from below. And so, from this study we found two important things. 1. Diamondback terrapins don't have Cloacal Bursae, and therefore can't use them to regulate gas exchange during hibernation, and so further research to understand how diamondback terrapins hibernate is required. And 2. Hatchling terrapins can't control their buoyancy and are therefore more exposed to predation than yearling turtles. And so that's all that I have for you today, and I'd love to take your questions now.